Welcome to Biochemistry DSP channel. In this video, I'll be explaining about the metabolism of very low density lipoproteins VLDL. Very low density lipoproteins. This VLDL are synthesized in the parenchymal cells of the liver. It contains apoprotein B100, apoprotein C and apoprotein E. This VLDL is composed of 95% of lipids and 5% proteins. Uh, the lipid content of this VLDL will include 60% will be of triglyceride and 15% phospholipid, and it will also have 20% cholesterol. So this is the composition of lipid components present in VLDL. It contains triglyceride 60%, phospholipids 15%, and 20% will be cholesterol, and the remaining 5% will be of protein. So the apoproteins we can find on the on VLDL is apoprotein B100, apoprotein C, and apoprotein E. This corresponds to the 5% of this protein. This VLDL is synthesized in the parenchymal cells, as well as it is also synthesized. A small amount will be synthesized in the intestinal mucosal cells. This VLDL will be released in blood from hepatocytes through reverse spinocytosis. The nascent VLDL in liver will contain only apoprotein B100. Okay. While entering into the blood circulation, it will acquire ApoE and ApoC proteins from the circulating HDL. Now we'll go into the metabolism of VLDL. So when the VLDL reaches the peripheral tissue, the apoprotein C2 will activate the enzyme lipoprotein lipase, which helps in the liberation of free fatty acid, and these free fatty acids will enter into the extrahepatic tissues for further metabolism to provide energy. So this VLDL or the chief carrier of this endogenous TG triglyceride. So endogenous triglyceride means whatever triglycerides which is synthesized in the liver, and helps to transport this endogenous triglyceride from the liver to the extrahepatic tissues, that is the peripheral tissue. The extrahepatic tissue means it includes adipose tissue and skeletal muscle. So in these tissues, the triglycerides contained in the VLDL will undergo hydrolysis by the action of the enzyme called lipoprotein lipase. So this lipoprotein lipase will be activated by the apoprotein C2, and insulin also will activate this enzyme lipoprotein lipase. So by the action of this lipoprotein lipase, 90% of TG will be converted into glycerol and free fatty acid, and these free fatty acids will be taken up by the tissues. After the removal of 90% of TGs from VLDL, it will be called as either IDL, that is intermediate density lipoprotein, or it is also called as VLDL remnants. Okay, the size also will decrease here, and the uh, density of this VLDL will increase due to the presence of this cholesterol rich. It is having now, it is rich in cholesterol. Now it will leads to the formation of this VLDL remnants, and these VLDL remnants will contain only the apoprotein B100 and apoprotein E. It will not have apoprotein C. So this is also called IDL, which will lack apoprotein C. Thus released apoprotein C from this VLDL, mature VLDL, will be used up to form HDL, okay, high density lipoprotein. Then this LDL. Okay, now from this IDL, it will be used up. This from this IDL, we can it will be used up to form LDL. So when you find a VLDL, it will be having richest source. It will have most 100% of TG and cholesterol, with apoprotein B100, apoprotein E, and apoprotein C. So after the action of this lipoprotein lipase, it will be converted into glycerol and free fatty acids. So most of the TG will be converted into free fatty acids. Now it will be left in the form of IDL, intermediate density lipoprotein, or VLDL remnants, which will lack this apoprotein C. Okay, it will have only apoprotein B100 and apoprotein E. The fate of this IDL or VLDL remnant is it will be uptaken by the liver cell. So here this VLDL will be acting as the precursor for IDL formation. Okay, IDL in turn, IDL will be the precursor to form LDL, that is low density lipoprotein. If you see low density lipoprotein, that 10% TG also will be removed. After the removal of, after the hydrolysis of this TG, only cholesterol alone will be left. That part with cholesterol with apoprotein B100 alone, with the release of E also will be released. Okay, that is called as low density lipoprotein, LDL. So VLDL will be acting as the precursor for IDL. IDL will be then converted into LDL. Only one molecule of it will contain only one molecule of apoprotein B100. Why means each LDL particle will be derived only from one VLDL particle. That's the meaning. Okay, so you derive one LDL particle from only one VLDL particle, so it will contain only one apoprotein B100. Then the for this ideal there will be two possible fates. Okay, it can be either converted into LDL, that is one fate for this ideal, or it can enter into this liver cell through LRP. Okay, through receptor, that is LDL receptor, which are specific for ApoB100 and ApoE receptor. So through LDL receptor mediated endocytosis, or through this only this LDL mediated receptor will allow this IDL to enter into the liver. Okay, so through LDL receptor mediated endocytosis, it will be converted, it will enter into the liver cell, where triacylglycerol and cholesterol will be further degraded, or the other fate for this IDL is it can be converted into LDL, and it which can enter into extrahepatic tissues. So this is about the metabolism of very low density lipoprotein. Then thereafter the metabolism of LDL will take place. Okay. Thank you for watching this video. Subscribe and support the channel Biochemistry DSP.